I'm Allison Burrell with Radiation Research Society Vodcast, and I'm here with Sebastian Oeck from Germany. What institution are you with?
Yeah, so I'm from West Germany, from the Institute of Cell Biology. Mm-hmm. It's related to the University Hospital in Essen, and the University Hospital as a teaching hospital is part of the University Duisburg-Essen.
Great, and your research is on a specific mutant protein that, is it a radiosensitizing, or what does this mutation?
So, I mean, the Protein Kinase B, this is my protein, also known as AKT. Actually, it's a major switch in the cell, dealing with survival, dealing with proliferation. Mm-hmm. And this mutation I'm working on mainly is enhancing proliferation in first step, but also, and that's what I'm doing during my PhD thesis, I've seen that it is also enhancing radio sensitivity, and yeah.
So proliferation of cells, you're working on a cell model?
Cancer cells. Right.
Okay.
Mm-hmm.
So you're saying it increases proliferation.
Mm-hmm.
But isn't that the opposite of what you want to happen with cancer cells?
Yes, for sure. So the mutation we are looking at is a clinical relevant mutation. So we try to target patients or tumors in patients with carrying this mutation, so that we are able to sensitize the cell and even, for example, impair the proliferation by hitting the AKT, especially the mutated AKT. Mm-hmm.
Okay, with radiation.
With radiation and also other components like small molecule inhibitors, to get rid of these positive effects on proliferation, on radio resistance of this mutation.
Okay. So what types of cancers do you find this mutation in? And it's naturally occurring, I guess.
So the special point mutations, so we're just talking about one amino acid, you mainly find them in brain tumors, but you also find it in bladder and prostate cancer and breast. But it's not that high occurring in, so we are not talking about the PI3K mutation, where you have about 30% in breast cancer.
So this is a mutation that doesn't occur as often, but could be exploited in precision medicine. So finding a patient who has this and then designing the treatment around your knowledge of what this mutation does. And is it an early mutation in cancer, or does it happen later in the progression of cancer?
So far, I'm not totally sure if there's really a cancer just based on this mutation. So there are many cases that have KRAS mutated or PI3K mutated, and some of them are carrying also the E17K mutation.
So it might be a later event.
Might be, yeah.
As it starts collecting mutations.
But so especially in my project, we are also using this mutation as a model for an activated AKT kinase. So also in tumors that have a PTEN loss, for example, or a high active PI3K, you get a high active AKT as a result, and we use this mutation as a model for this.
Okay, so is there, so how do you use this mutation to, you know?
So what we did was we introduced via retroviral gene transfection, stable expressing these mutants in murine cell lines. And so we want to try a kind of a chronic model, like you have it in a tumor. You have a occurring mutation that is always there. And if you use a transient system, it might get lower, or it is thrown out, or whatever, and the expression goes down. And with this retroviral system, we were able to have all the time same level of the protein. We can compare it to the AKT wild type, also expressed with the same model. And so you have always a good control for this system.
Okay.
Yeah.
So then what's the next step after you introduce this mutation in the cell lines?
So basically, we were using ionizing radiation, X-ray, and check for survival, proliferation upon radiation. And in the next step, we also took a look at the DNA repair. And always in context, we check for localization of this protein, phosphorylation state, phosphorylation of downstream targets, and stuff that is actually related to this protein and the whole pathway.
All right. I guess what kind of assays are you using to look at the DNA damage?
So we have direct and indirect assays. Indirect assays, the common used gamma H, for example, so immunofluorescence. Also, we were taking a look at specific repair pathways like the homologous recombination repair with RAD51 foci creation, for example. And, but we also used direct assays, so COMET assay, for example, so that you can see also real damage on the DNA. And I mean, if you take a look at a high active protein kinase, it might be that there are other pathways changed somehow. And if you then just take a look on a phosphorylation or on accumulation of foci of different proteins, it might be not enough to check. Mm-hmm. So and that's why we, in the later part of my project, we go on with this direct assays. And, but actually we confirmed the experiments, so we confirmed the results of the gamma H assay and everything, so this was quite nice.
So what's the clinical application for this?
So in the end, I think it might be possible to sensitize especially tumors carrying activating mutations of the PI3K-AKT pathway, so that you really can sensitize them for ionizing radiation. And that might be better also if you think about normal tissue effects, that you have really a target that is on the one hand, for responsible for the proliferation and the tumor growth, but that is also doing something with the radio resistance. And if you hit just one target and get rid of it, of the proliferation and the radio resistance, this might be very nice.
What kind of delivery system are you thinking of? I mean, this sounds like gene therapy, or?
So there are some clinical trial 2 inhibitors of AKT, like the MK2206, for example. This is ongoing work of other groups also.
Are these already medications that are FDA approved?
Not yet, totally, okay. But I think they're on a good way. But there are some already in use, I think also in the clinic, like PI3K inhibitors. And it might be that they are also working, so we tested also PI3K inhibitors. And yeah, this might be also a way to radiosensitize the cells.
That would be really interesting. What are your next steps, I guess?
So my next step will be to spend a year in the US doing some research. So I finished my PhD thesis, and I think this, for me, was so the main part of the work with this project. But there's a follow-up project in Germany, and they are now a little bit more looking at specific targets upon radiation of the protein kinase, so that we maybe can hit a little bit more specific on this radio-resistant effect by hitting a target of the AKT.
And so that would be done in a mouse model?
Yeah, but we're also trying now to do xenograph with human cells. So.
Right, because you were using murine cells to induce.
Mainly because of the retroviral system.
Right.
Yeah.
Okay. So you didn't use any human cells?
Not yet.
Not yet. Okay. Well, it sounds really interesting, and it's always good to have like a combination, something that you know that's sensitizing the cells, and then giving the radiation treatment. are you at all concerned about heterogeneity of the tumor? Not all of the cells having this mutation, using this inhibitor might sensitize some of the cells, but then the others are still going to survive and keep proliferating?
So on the one hand, it's really one of the big difficulties in my project that AKT is just everywhere and always working and doing something. But on the opposite, this is the nice effect because you can always inhibit it.
Right.
So, but of course you have to be careful of normal tissue effects. If you just put in an AKT inhibitor, you might get problems in proliferation tissue.
Yeah, I'm wondering what the consequences or side effects are of inhibiting AKT in a normal cell, or a cancer cell that doesn't have this mutation of this specific amino acid. Are there other AKT mutations that, like besides this one amino acid, that cause this effect?
So we did a lot of database analysis of tumor samples, and I'm pretty sure that this is one of the only so far found that is really in a somatic way.
How big is the protein? How many amino acids?
We are talking about 480 amino acids about. So this comes out in a 60 kilodalton protein. And what the mutation is doing actually is enhancing the binding to the membrane via the pH domain of the AKT. And so it is quite at the beginning of the whole pathway, directly after the PI3K. So this might also be an interesting point that you have a very early activation, very upstream in the pathway. And on the opposite, this is one of the reasons why we are now looking at downstream targets also. So it might be that AKT it is a little bit more important where AKT is. So really that the localization makes a choice for AKT what kind of next downstream target will be phosphorylated.
Well, and I guess what's good about this is that pathway is well elucidated, so you know what downstream factors you can investigate next. Yeah, great. So you said this was your PhD project, and so you're leaving the lab now and won't be continuing with this. I mean, the lab will continue with this project, but do you want to stay working on something similar, or what are your ideas for your next step?
So I think I will not completely stop working on AKT and these mutations. I will now go a little bit more in the microRNA field, but also taking always a look at DNA repair, especially connected to ionizing radiation.
Right.
And I think we are now on a good project, trying to create a new project, and hopefully there will be some new plans coming out next year.
Great. Well, thank you so much, Sebastian, for talking with me. Is there anything else you'd like to talk about? No. All right.
Well, thank you very much.
Yes, thank you very much. Nice to meet you.